Hello, everyone, and welcome again to Biochem. Today, we're talking about the mechanisms, models, and the clinical significance of enzyme specificity. So, to talk about enzyme specificity, we need to talk about the active site, which is more like a crevice or a pocket to which the substrate binds for a chemical reaction to progress. And this molecular architecture of the active site is based on the geometric complementarity, the electronic complementarity, which has to do with the pH or the polarity of that has been optimized for the reaction to progress. For the electronic complementarity, it has to do with the charge that the substrate is carrying, and while the shape is also very important for the reaction to progress. So looking at the selectivity problem of enzyme, it is really not a problem to me because it's one of the things that makes biological reactions to be able to progress and be ordered. So the cell is crowded, has so many metabolites, so many reactions occurring simultaneously, and we have different molecules of different enzymes catalyzing different molecules. So what this shows is that the orderliness is maintained as a result of the three-dimensional recognition of the active site of enzyme to be able to pick what molecules it's able to act on. So looking at the precision microenvironment of the active site, which is based on the size, the shape, as I mentioned, the charge and the hydrophobicity alignment between the active site and the substrate itself. And this is dependent on sometimes the alpha helix beta shapes sheets that forms the scaffold of the enzyme active site itself, and sometimes the presence of cofactors like NADH and some metal ions that are present for the biological reaction to be able to progress, as well as sometimes the presence of water. Now, looking at the spectrum of specificity, it ranges from absolute to group. You have absolute, which is one-on-one, one substrate, one enzyme. You have group, where you have one enzyme being able to cater to a group of substrates based on the presence of the R group. And then we have the bond or the linkage, where the enzyme is acting based on the type of bond the substrate carries. And we have the stereo, identify the enantiomers, the mirror image, which one, the L, the D. And the last is the reaction. For example, the kinases being able to phosphorylate. So that's the type of reaction kinase catalyzes. And we have oxidases based on the oxidative reaction. So this is more like a pictorial representation of how this specificity spectrum cut across. There were some hypotheses that were trying to explain this enzyme specificity and how reaction progresses. And one of it is the lock and key model, which defines the rigid complementarity of enzymes, saying that substrate is able to fit into the active site of the enzyme in a lock and key model. So there's usually, this explains that there is usually no conformational change. It just has to be a perfect fit into that rigid structure. And so then it will progress from the fitting of enzyme substrate complex into enzyme product complex, and then to the final model. But this explanation has a limitation in the sense that it fails to explain what happens at the transition state. So this led to a more newer model that was released, and that is called the Induced Fit model, which explains the dynamic conformational change that happens at the active site, saying that when the substrate binds to the active site of the enzyme, there is a dynamic shift in the shape of the enzyme, allowing it to suit the conformation that the substrate needs to be able to trigger that reaction, progress to the next reaction phase. So the active site shapes around the substrate, and then it creates this tight fit for the reaction to progress. So this distorted geometry of the enzyme substrate complex strains the substrate towards its transition state. So that dynamic shift, that geometric distortion strains the substrate into its transition state. And this is what lowers the activation energy that is required for the reaction to progress, then speeding up the rate of reaction. So one of the reasons why enzyme is faster and more powerful than the simple chemical catalyst is because of this ability to manipulate or cause a dynamic shift in the structural component of the substrate it binds towards releasing the product. This is just like a difference differences identified in the two models that explains the enzyme specificity. Now, let's pick the absolute specificity and try to zoom in. So a common example for this is urease, which acts on urea and leading to the release of ammonia and carbon dioxide. So urease is only specific to its substrate urea. If you present it with methyl urea or hydroxyurea, it will not act on it. And this is very important, critical for nitrogen metabolism. So another very common one, which is group, as I mentioned earlier, is able to identify the R group. For example, in the case of alcohol dehydrogenase, it can act for propanol, it can act for methanol, it can act for ethanol, and that is because of the presence of the hydroxyl group, stereo isomer, the stereo identification. The active site is able to identify chiral centers. So it's able to detect this geometric or the spatial position of a substrate, not just the chemical nature. It's also able to detect the three-dimensional arrangement of the atoms that are present in the molecule. And so when there is no fit, even when the shift comes, then the reaction will not be able to progress. And this is what allows an enzyme that is specific for L-adenine, L substrate or the level substrate, not to interfere in the catalysis of a D substrates. So as in the example that you can see on the screen, L-alanine is acted upon by a specific enzyme that recognizes the level rotatory image, while D-alanine is acted upon by the specific enzyme that recognizes the dextrorotatory image. This chiral recognition for activity of the enzyme is what help it with that inherent asymmetric recognition of its environment. So specificity can also be toned down to look at promiscuity. So while it's important that we consider that specificity is not always exclusive, under some conditions, some enzymes are able to catalyze more than one reaction, or even beyond their primary function that they are known for. For example, if an enzyme is known to catalyze a particular R group, sometimes they get into some secondary catalysis, and that is what this promiscuity is all about. For example, we have glycolytic enzymes being able to switch sometimes to act as an oxidative reductase. And so as you can see in this example, you have glycerol phosphate dehydrogenase, which catalyzes the activity, the conversion of glycerol phosphate to 1,3-bis phosphoglycerate in the glycolytic pathway. This is in normal glycolysis, leading to energy generation. This promiscuity also now come into play when it is acting under an oxidative stress situation. When the condition change, it's more like an adaptive response, and then it is converting glycolytic, and this same glycolytic enzyme is acting as an oxidative reductase, converting your the sulfurhydrate group of the protein of a protein in the presence of a reactive oxygen species to disulfide bond in the protein. And so it's acting as an oxidative reductase to protect the cell from some sort of oxidative damage, but you can do more digging. So if you have a better explanation, please, you can drop it in the comment section. Substrate promiscuity, when it's able to pick on different things like the way GST is able to metabolize hundreds of xenobiotics. And we have catalytic promiscuity when we have serine proteases being able to act on esterases and also act on ligases. So the next one is this condition promiscuity, when we have same enzyme, different reaction when in water versus when it is in another organic solvent. The last one is evolutionary relevance, where you have glucuronidase becoming beta-galactosidase as a result of its mutation, like 8 mutations or so, catalyzing different reactions. So the clinical application has to do with the drug metabolism promiscuity in cytochrome P450 during drug-drug interaction, and as it relates to the cytochrome 3A4's broad spectrum tolerance. So now let's look at, we all know that AI is at the frontiers of so many things now, and we want to see what AI is doing in this space as it relates to biochemistry of the enzyme specificity. So AI is able to predict, just as we are trying to learn about enzyme specificity now, the enzyme specificity and data has been presented to AI for it to be able to learn what is happening with enzyme specificity, and it now automatically knows what is happening with it, how the dynamics works and its mechanism. Now, one of the identified one is EZ specificity, which is I think is a 2025 tool that has been discovered to take two inputs. So what it does is to take the enzyme's 3D shape and the substrate's chemical structure. And so when it takes this, it does some cross attenuation. When this is fed into an AI model, it does some cross attenuation, looking into the chemical structure, the substrate's chemical structure, and then it comes out with an output of a simpler answer, letting us know if the enzyme will be able to catalyze that molecule. If it is yes, how energetically favorable is it? Talking about the Gibbs free energy that is involved for the biological reaction to progress. And so when EZ specificity came, it hits around 91.7%, which is quite good. And in practical terms, what this means for us is that we can now screen thousands of enzyme substrates pairs. So if I have an enzyme that I suspect I should be able to do this, and this is helping a lot in disease, drug discovery, and also in understanding disease mechanism computationally. So within hours, we are able to predict what the drug side effect will be. And then we're able to also, researchers are able to find new antibiotic targets that they can work with. So I see that without even having to run a single lab experiment, you might be able to predict some movement. So in summary, we look at the three-dimensional recognition of the substrates by active sites, and then we looked at the different groups. We talked through absolute, group, bond, talking about the linkage as well as the reactions spectrum. And then we looked at the active site triad, looking at that pocket, what are the things that are responsible for the function to be able to progress. So, and then I think we also touched on the model, lock and key versus the induced fit, what were some of the limitations that they had. And the last one was the stereo, looking at the enantiomers, the mirror images, how they're able to identify themselves, and the promiscuity, which is more like the other side of specificity, being able to look into some of the functions that enzyme could perform outside its primary function. And the last is the AI model, what AI is doing, being able to understand and being able to use this in clinical application. So thank you so much for joining me on this class. Please put your comments if you like this video, if you find it interesting, and please share with your classmates and other people that may be learning this, and subscribe to my channel so that when I get a post, you see it. Thank you so much for